There are times in genetics when the genotype of an organism is to be determined. That is, you don't know if the organism is homozygous for a trait under consideration or heterozygous for that same trait under consideration, for which it expresses a dominant phenotype. In order to do this, a test cross is carried out. What is a test cross? A test cross is a cross between an individual who is homozygous recessive for a given trait and an individual whose genotype for that same trait under consideration is unknown. For instance, in this case, we don't know the genotype of this red colored flower. We don't know if the red colored flower is homozygous for that trait. That is, if it has the same allele represented with capital letter R, or heterozygous for that trait. That is, it has different allele, capital letter R, small letter r. What we need to do is to cross this plant whose genotype is unknown with the recessive form of that plant, and the recessive form of that plant, which is homozygous recessive for the color under consideration, is white. So from the offspring produced, we can determine if the genotype of that plant is homozygous or heterozygous. If all the offspring that are produced from the crossing expresses the dominant character, that is, all the organisms are all red in this case, that organism is said to be homozygous for that trait under consideration. But if half of the offspring that were produced are white and half of the offspring that were produced are red, therefore the organism is said to be heterozygous for that same trait under consideration. In this case, let's assume this organism whose genotype is unknown is homozygous, that is, has the same allele, capital letter R, and it was crossed with a plant who expresses the homozygous recessive trait, which is white, small letter r, small letter r. For genetic crossing to take place, there is need for gamete production. So during gamete production, each of these genes goes into the gamete separately, and same goes for the white plant. Then genetic crossing takes place. This gamete, which contains the gene for red color, crosses with this gamete to produce capital letter R, small letter r. So this offspring is said to be heterozygous for that trait. Why? Because it contains the gene that shows the dominant character, which is color red, and at the same time contains the gene which shows the white color. But in this case, this small letter r is recessive. This same gamete also crosses with this gamete to produce another heterozygous offspring. This also crosses with this to produce another heterozygous offspring, and this crosses with this to produce heterozygous offspring. So all the offspring here shows the dominant character. All the offspring in this case, they show the red color, but they are heterozygous for this trait that is under consideration. And don't forget that I said if all the offspring that are produced they all express the dominant character, that shows that the genotype of the unknown organism under consideration for that given trait is homozygous. So in the case of this red colored plant, it is homozygous. Let's look at a situation where the organism is heterozygous for the trait. If the organism is heterozygous for the trait, from the offspring produced, you will know, because half of the offspring will show red color, which is the dominant character, and half of the offspring will also show the white color, which is recessive for that same trait under consideration. So let's look into this. Gametes are produced, and from the gametes produced, genetic crossing takes place. So this crosses with this to produce capital letter R, small letter r, heterozygous red. That is, it has the gene that codes for red color, which is the dominant gene, and at the same time has the gene that codes for the white color, which is the recessive gene. This same gamete also crosses with this to produce heterozygous red. This one, which is the gamete that has the gene that codes for the white color, which is the recessive trait, crosses with another gamete that houses the gene that codes with that same color white to produce small letter r, small letter r. So this organism is homozygous recessive for the trait under consideration, and this also goes for this to produce small letter r, small letter r. So from the offspring, half of them are red, half of the offspring are red, heterozygous red, and half of the offspring are homozygous white. They are recessive. So this organism is said to be heterozygous from the trait. In order for you to know if an organism is homozygous or heterozygous, information from the offspring will determine if all the offspring are said to express the dominant character, then the organism is said to be homozygous for the traits under consideration. If half of the organism shows the dominant character and half of the organism shows the recessive trait under consideration, then the genotype of the organism under consideration is heterozygous for that same trait.